We are going to continue with our series of lectures on the digestive system. I think this is the third part of it. There's still one or two more to go. Today, we'll be looking at the organs, the extrinsic organs of the digestive system. But just to remind us that we mentioned that the digestive system consists of the part followed by food, which we call the holoGIT, and the extrinsic organs of the digestive system. These are organs which are not within the wall of the GIT, but provide their secretions into the lumen of the GIT. And so today we are going to focus on these four categories of organs. In our lecture outcomes, we've already talked about the components of the holoGIT proximal-distally. We've also talked about the histology of the gut wall. We've described the anatomy of the different parts of the gut, basically. And so today we are at objective number 4, which you're going to do. We're also going to achieve objective number 5. Let's start with that objective number 4. When you talk of extrinsic glands or organs of the digestive system, we are referring to the salivary glands, the liver, the gallbladder, or if you want to call it gallbladder, and the pancreas. Let's start with the salivary glands. We have two categories of salivary glands. There are those that we call major salivary glands and the minor salivary glands. The major salivary glands are the ones which qualify to be called the extrinsic glands of the digestive system. This is because they're the ones which are not within the wall of the holoGIT. The minor salivary glands are intrinsic glands. They are within the wall, and so they're not extrinsic, but either way, they're still salivary glands. So which are these major salivary glands? We have the parotid gland, we have the sublingual gland, and the submandibular gland. We will talk about them shortly. But just to mention, the minor salivary glands are the ones located within the wall of the mouth, basically. So there are those that are in the palate, you call them palatine glands. The ones in the cheeks, you call them buccal glands, and the ones in the tongue are called lingual glands. They all produce saliva, whether major or minor. What does the saliva contain? Saliva contains many things. Of course, there's water. This water has digestive enzymes. In specific, we have ptyalin, which we also call salivary amylase. This is the one that breaks down carbohydrates. We also have the salivary lipase, which is sometimes known as the lingual lipase, the one that breaks down lipids. Then we have mucus. Mucus is a lubricant, basically. There are some minerals within saliva, and there are also some chemicals which are helpful in fighting infections within the mouth. So histologically, how does a major salivary gland look like? Major salivary glands are lobulated glands. So as you can see, this is one lobule, that's another lobule, that's another lobule, that's another lobule. They are lobulated glands. Between the lobule are connective tissue trabeculae, like that region. And within this connective tissue trabeculae, you may have blood vessels, as you can see here. The connective tissue trabeculae are between the lobules. So this one lobule, this is a trabecular system. At a high magnification, we recognize a number of things which are within the parenchyma of a salivary gland. The parenchyma of a salivary gland contain histological structures organized in form of acini. Acini are basically exocrine glands. So this is an acinus. This is an acinus. The plural is acini. There are two types of acini in a major salivary gland. There are those acini which do not take the stain, as we can see in this one. Those ones are called mucus acini. To mean that the secretions they contain are mucoid, and so they don't take this serous, they don't take this hydrophilic dyes that we are using to stain the cells. So they don't take the color up. These are the mucus acini. But you also have the serous acini like this one, or that one, or that one. The acini which take stain, the ones which pick color, they are picking color because their contents are basically hydrophilic. And so they're able to take the colors that we use in histology. We call them serous acini. So there are two types of acini in a major salivary gland: mucus acini and serous acini. The acini are the ones which secrete mucus. Sorry, the ones that secrete saliva. So it could be saliva that is rich in mucus, very thick saliva, or it could be saliva that is not very rich in mucus, thin saliva. And I believe you're familiar with those two types of saliva. Because they are exocrine glands, they must have duct. And so the parenchyma of a salivary gland also contain a ductal system. And these are the examples of ducts that we see that carry saliva away from the gland. Usually, a major salivary gland will also have some scattered adipocytes. Nothing much to say about them. They're just fat cells which are scattered. So they don't really form an adipose tissue within the gland, not quite. They're just scattered cells. And that shouldn't really raise a lot of eyebrows. Adipocytes are usually scattered in almost all parts of the body apart from the brain and the spinal cord maybe. Usually adipocytes are found many places. Even some parts of the brain actually may still have the adipocytes, except that one will be in a context of a malformation. So I want us to narrow down to specific major salivary gland, and I'll start with the parotid gland. The term parotid comes from two terms, parallel and otid. The otid refers to the ear. Parallel means on running aside or something. So parotid is a gland next to the otid. And that's why it's called parotid. It's a gland next to the ear. The parotid gland is the largest salivary gland. We know that on either side of the face, basically anterior to your ears, usually extending slightly down. And from your head and neck anatomy, you remember that the parotid glands are just below the zygomatic arc. The duct of the salivary gland is a single duct that drains into the buccal cavity, specifically in the vestibule of the mouth, opposite the second upper molar tooth. So remember, it doesn't drain into the oral cavity proper, but it drains into the vestibule of the mouth, opposite the second upper molar tooth. Usually, if someone retracts, you can actually see the opening of the parotid duct, of course on the cheek side, not on the gum side. Parotid gland is primarily innervated by the glossopharyngeal nerve, albeit the fact that the facial nerve is the one that passes through parotid gland. The facial nerve does not really supply parotid gland for secretion. For this gland to secrete saliva, it requires stimulation by the glossopharyngeal nerve. And this innervation is primarily parasympathetic innervation. So glossopharyngeal nerve is parasympathetic to the parotid gland. And parotid gland produces a lot of serous secretion. It is the secretion from the parotid gland is predominantly serous. And this should make also a lot of sense. Remember, parotid gland is the gland that is farthest away from the oral cavity. So it can't afford to have a lot of its secretion being mucoid, because that will have difficulty traveling along its duct to the mouth. It will take forever, or the risk of impaction will be a lot. So usually, as a concept, the further a salivary gland is away from the mouth, the more watery its secretion would be, to ease the probability of transportation of the saliva to the mouth. So histologically, how will we pick a parotid gland from any other salivary gland? First, at this magnification, we appreciate that, yes, we are looking at an organ that has a capsule, and you can see the connective tissue capsule outside there. Then within it, we can appreciate that, yes, there's some level of lobulation. You can see a lobule there, lobule there, lobule there, lobule there. With the trabecula, even though they are tiny at this magnification, because I'm looking at very low magnification. But you can see lobulation somehow. You can also see scattered adipocytes. Those things which look like empty spaces, those are scattered adipocytes. And you can also see a ductus system. These are duct, that's a duct, perhaps that's a blood vessel, but these are duct within a thick connective tissue zone. So you see duct, you know that this is most likely an exocrine gland. Perhaps it would be difficult for you to say it's parotid at this level if you haven't seen much of them. But at this magnification, we're able to now see that the parenchyma of this gland consists of predominantly serous acini. So we can see the acini there, they're predominantly serous, which means they are picking up the stain. And you can also see duct. So we see serous acini and we see ductus system. We almost narrow it down to the parotid gland. Of course, in the context of the scattered adipocytes also, we know that this is basically a salivary gland, and parotid gland is the one that is predominantly serous. I'm saying so because you're going to see the other two very different from parotid gland. At a high magnification, this is just showing you how serous acini look like. These are serous acini. You can see the way it is taking up the stain. That's another acinus, that's another acinus, that's another acinus. And these are the ducts. Those are the adipocytes at a high magnification. All right, this one shows us at least the capsule, it's encapsulated. It's also showing us the different types of ducts there. I want us to talk about the ductal system of the parotid gland. I've mentioned to you that any salivary gland is lobulated. The major ones that are, they are lobulated. So we have connective tissue that separate the lobules. There are some ducts which are within the connective tissue that separate the lobules, like this duct here. The ducts which are within the connective tissue that separate the lobules are known as extralobular ducts, which means ducts outside the lobules, extralobular ducts. And that is to distinguish them from the ducts which are within the lobules, like that one or that one or that one, which we now call intralobular ducts. Your guess is as good as mine that the extralobular ducts are larger, as you can see even here, and that they're the ones that receive the intralobular ducts. So the intralobular ducts are within the lobules, and they're the ones that join and drain to the extralobular ducts, which are larger. As you can see, the extralobular ducts are within a mass of connective tissue surrounded by some blood vessels, as you can see here. Let's say something about the intralobular ducts, the ducts within the lobules. There are two types of intralobular ducts. There's this one here. Maybe you're struggling to see it, but now that I've pointed at it, you can see it. There are two types of intralobular ducts. This one here is known as the intercalated duct. The intercalated ducts are the initial ductal portions which arise from an acinus. And that's why you might even find it difficult to pick that out easily, because we're just seeing something very close to an acinus, but not an acinus. If you look at the cells there, they're a bit different from the cells of the acini. So this is called the intercalated duct. It represents the initial ductal segment that arises from an acinus. To distinguish it from this one here, which we call striated duct. So a striated duct would look like that. And why do we call them striated? I'll give you a high magnification of it. So this is the high magnification of a striated duct. You'll see its whole wall is complete basically, but I want you to look at the basal zone of the cells, the region external to the nucleus of each cell, and you see some very vague things which look like lines perpendicular to the base, and these are the striations, like alternating dark and light bands in this region. That is why we call them striated. These striations are not because of alterations of actin and myosin, as maybe your mind is already running towards, not quite. It's about the arrangement of mitochondria within this region. These ducts have several mitochondria arranged in columns on the basal side. At light microscopy, they can just appear like lines. At electron microscopy, they can be visualized better. These mitochondria tell us that these ducts are highly absorptive, or rather, they're highly active because the mitochondria provide energy. Now, the activity is largely for ion transport. These cells are adapted for active ionic transport. And so that's why they have the mitochondria to produce the ATP necessary for transport of ions. Remember, we have extralobular ducts and interlobular ducts. The interlobular ducts, you have intercalated duct and striated ducts. So the intercalated ducts drain to the striated ducts, and the striated ducts drain into the extralobular ducts, which then drain into the major parotid duct, which go all the way to the vestibule of the mouth opposite the second upper molar tooth. The second salivary gland to talk about is the sublingual gland, located below the tongue. That's why we call it sublingual. The sublingual gland does not have a single duct per se, but multiple ducts, tiny ones. They drain on the floor of the mouth. Usually, if you elevate your tongue, you'll be seeing an elevation of the sublingual gland. You'll not be able to see the ducts because they're very tiny, microscopic, multiple of them. Sublingual gland is innervated by the facial nerve, and sublingual gland produces mucoid saliva. Now, the concept I was telling you should now click home. Sublingual gland is near the mouth, if not within the mouth, of course. And so the secretions are very thick. There's less distance for the mucus to travel anyway, so not a big deal. Histologically, this is how the sublingual gland will look like. So several concepts we've already underscored when we talked about parotid gland. The main thing I want to highlight is that the parenchyma of the sublingual gland will now be made up of predominantly mucoid acini instead of predominantly serous acini. And you can see what I mean by predominantly mucoid acini. You hardly see any serous acini in this field. Well, I chose it intentionally to just bring out the message home that it will be predominantly mucoid, but you may still have some serous acini within the sublingual gland. Lastly, we have the submandibular gland. The submandibular gland is below the body of the mandible. That is why it derived its name as such. So this gland has a duct. The duct of the submandibular gland drain also into the floor of the mouth. Usually, if you lift your tongue, you'll see that midline thing, which we call the frenulum of the tongue. It's like the one that holds the tongue down. On either side of the frenulum, you may be able to see some things which look like the tip of a pimple. Basically, those are the submandibular duct puncta, the openings of the submandibular gland duct, the duct of submandibular gland. These glands are also innervated by facial nerve, just like the sublingual gland. And the submandibular gland has mixed saliva. The saliva has a mixture of both serous and mucoid, perhaps at a near relationship. It's not necessarily 50-50. It could be 40-60 in terms of the proportion of what is mucus and what is serous. So this is how a submandibular gland would look like. At face value, you may think this is parotid because you're seeing ducts, yes, you're seeing lobulation, yes. You are seeing mucus acini, you're seeing serous acini. The mucus acini at this magnification may confuse you for the scattered adipocytes, and so you might want to call this parotid. But at a higher magnification, you'll appreciate that they are not adipocytes. They are mucus acini, and you can appreciate that in this view. So, but the fact that you have mucus acini and serous acini mixed, it means you're looking at a mixed salivary gland, and submandibular gland is the best shot for this one. Otherwise, the other things still stand out. We have ducts which are interlobular and extralobular. Something very unique about the acini of the submandibular gland is this. There are some regions where you have a mucus acini, like that one. But on the periphery of that mucus acini, we have a serous cell, like this one. This serous cell somewhat look like the crescent moon around the mucus acini. You can appreciate them even on this one, or even on this one. We have some serous cells. Serous means they're staining. Serous cells which form something like a crescent moon on the periphery of a mucus acinus. Those cells constitute what we call the serous demilunes. Demilune means half a moon. The submandibular gland is unique with this feature, that it contains serous demilunes around the mucus acini. You do not see that one on any other salivary gland apart from submandibular gland. The presence of serous demilunes. They are serous cells which are on the periphery of mucus acini. We are done with salivary glands. I want to pick the next extrinsic organ, and that is the liver. So the liver is located in the right hypochondrium as well as the epigastrium. Usually the right lobe of the liver will be the one in the right hypochondrium, and the left lobe of the liver will be in the epigastrium. And we know that the liver is below the right hemidiaphragm predominantly. Anatomically, there are two key lobes of the liver, although sometimes we talk about 4 lobes of the liver. The 2 lobes of the liver are the right and the left lobe. In this image, we can see this the right lobe and this the left lobe. This shiny surface of the liver is called the diaphragmatic surface of the liver. It is the part of the liver that is related to the thoracic diaphragm. So we call the diaphragmatic surface of the liver. If you look at the diaphragmatic surface of the liver, we are only able to appreciate those 2 lobes of the liver, right and left. The right one is usually bigger than the left. However, if you flip the liver to the other side, we look at the side of the liver that faces the abdominal cavity, which we call the visceral surface of the liver. We see more than 2 lobes. There's this one here, which we call the caudate lobe, and there's that one there, which we call the quadrate lobe. This anatomy is better appreciated practically, and so there's a lot on this lobar anatomy of the liver that I'll not talk about today, but when we come for the practical, we will then appreciate at greater length. Then at that time, we'll tell you that we have right and left anatomical lobes, and with that classification, the caudate and the quadrate lobes are on the right lobe. But we also have right and left physiological lobes of the liver. And on that note, the caudate and the quadrate lobes are on the left physiological lobe. But in a nutshell, the anatomical lobe is based on the presence of this thing here, the falciform ligament, the level of the falciform ligament, which separate the liver into right and left anatomical lobe. The physiological lobes of the liver are based on the blood flow into the liver. So that we'll revisit practically, not in this virtual class. And we'll see how the liver receives blood supply. And we'll go ahead to even break down the physiological lobes of the liver into what we call liver segments. A total of 9 liver segments. So that after that, we'll be knowing when we say segment 1 of the liver, what do we mean? Segment 4B of the liver, what does it mean? Segment 8 of the liver, what does it mean? A total of 9 segments, named segment 1 up to 8, with segment 4 having A and B. So they have a total of 9. And usually this is segment 1, that's segment 4B. That's segment 2. That's segment 3. The rest, I think, we will talk about practically, not virtually like this. The visceral surface of the liver is worth mentioning because it's related to some organs. And I want you people to know the organs related to the visceral surface of the liver. So basically, this region here is the one that is related to the stomach. So we call the gastric area of the liver. The stomach comes like this, and the pylorus of the stomach lies somewhere here. So that's around the pylorus of the stomach still. At the stomach, the first part of the duodenum usually crosses like this. It may not make so good contact there, but the second part of the duodenum makes this impression here. So this is the D2, second part of the duodenum. Next to that duodenal impression is this region, which is the impression of the right kidney. So the right kidney makes this impression here. And above the right kidney, we have the impression of the right suprarenal gland somewhere there. This is the impression of the right suprarenal gland. Below the impression of the right kidney, we have the impression of the colon. So the point I'm showing you right now is where we have the hepatic flexure of the colon. And then around here, this is where we have the transverse colon. Don't confuse the impression, the transverse colon, with the impression of the pylorus. Of course, this is the gallbladder. This collapsed thing is a gallbladder. And so we can say this is a gallbladder impression. And this large thing you're seeing here is the inferior vena cava. And so in that groove, we can call the inferior vena cava impression. So there are many organs which are related to the visceral surface of the liver. There's this region in the liver, which we call the porta hepatis. The porta hepatis represent this part of the liver that receives blood vessels. There are 3 blood vessels which enter the porta hepatis, or rather, the part of the liver that receives some structures. There are 3 structures that go through the porta hepatis. They're not all blood vessels, my apology. There are 2 blood vessels and 1 ductus system. So what are these 3 structures which go through the porta hepatis? We have the portal vein, we have the hepatic artery, and we have the bile duct. So bile duct, portal vein, and hepatic artery. Those are the 3 structures that constitute the porta hepatis. And they tend to run like that even within the liver. Even at histological level, those 3 structures tend to run together. The name given to them is the portal triad. The portal triad represents the pathway of hepatic artery, the pathway of portal vein, and the pathway of bile. On that note, let's describe the blood supply to the liver. The liver receives 2 sources of blood supply. We have the portal vein, which carry deoxygenated blood from the intestines, as well as deoxygenated blood from the spleen. So the deoxygenated blood from the spleen, as well as the one from the GIT, join to form the portal vein, which again empty into the liver. And then you ask yourself, so why is deoxygenated blood entering the liver? Why can't it just go direct to the heart? And the answer is this deoxygenated blood contains nutrients and maybe even some toxins. That brings you to the functions of the liver. The liver needs to do something with those toxins, something with those nutrients. The portal vein contributes about 75 to 80% of blood flow to the liver. But because it doesn't have oxygen, the liver needs another source of oxygen. The source of oxygen to the liver is the hepatic artery. Hepatic artery is a branch of the celiac trunk, which is a branch of the aorta. So this hepatic artery delivers oxygen to the liver, responsible for about 20 to 25% of blood flow to the liver. Histologically, this is how the liver looks like. I know we will look at histology again, maybe next trimester. But at a nutshell, the liver histology is largely made up of what we call the histological lobules of the liver. There are lobules that you can see here, 5-sided, 6-sided, 7-sided lobules. Like these ones, we call those ones the classical lobules of the liver. As you can see, the classical lobules of the liver have something at the center, a blood vessel, which we call the central vein. I don't want to talk about the histological lobules of the liver today. We will do that again next trimester. But what I want to talk about are largely the cell types within the liver. Look at how the cells of the liver are arranged. They're arranged in somewhat cords. Those cells arranged in cords are the ones we call the hepatocytes, the major cell types of the liver. The hepatocytes are the ones that perform most of the liver functions. Apart from hepatocytes, we also have the Kupffer cells. Kupffer cells are liver macrophages. Then we have cells of Ito. Cells of Ito are fat-storing cells. They metabolize vitamin A. Then you have endothelial cells, the cells of the liver that line the liver sinusoids. Sinusoids are capillaries within the liver. So the endothelial cells that line those capillaries. And lastly, we have the cholangiocytes. These are the cells which form the Ductus System that contain bile. So they're the cells that form the bile ducts. These are the liver cells. And I want you to be familiar with these cells because the pathologies of the liver basically revolve around the cells. So there's some pathologies that will have hepatocellular as the main target. There are those that will target the cholangiocytes, there are those that will target the Kupffer cells, the endothelial cells, and the like. Let's make a summary of the functions of the liver, even as we finish on this one. I believe you'll do this again in physiology, and so I can just pass through this slide. The liver has metabolic functions. It metabolizes carbohydrates, lipids, and proteins, basically. It also stores some vitamins as well as iron. The vitamins which are stored in the liver are A, D, and B12. And iron is also stored in the liver in form of ferritin. The liver also synthesizes clotting factors. And the clotting factors which are synthesized in the liver are these ones: fibrinogen, prothrombin, factor 7, factor 9, factor 10. The liver also detoxifies and excretes some substances like drugs, hormones, even calcium. And some of these then are excreted through bile. There's some immunological function in the liver. This is principally the role of the Kupffer cells, which are the phagocytic cells in the liver. And the liver is a reservoir. Maybe this is not a function per se, but it has implications, and so it's worth mentioning that it has a reservoir function. The liver can hold large quantity of blood. And so in times of excess blood volume, it can hold large quantity of blood so that we still have enough for the circulation, not in excess. Okay, the third organ to talk about is the biliary system. Maybe first we identify the structures labeled A to D. I'm giving you 30 seconds to just name them. You can use the chat to just give names to any of them, structure A to D. Feel free. You can just write to everyone, not a big deal. And if one has been answered, you don't have to insist again. Carry on with the chat. Fair enough, carry on, carry on. Aya, tushajibu liver. Okay, we've gotten the pancreas as well. We've gotten duodenum as well. So what is structure boy? Fair enough, we are good. We are now okay. I've closed the chat so that you don't have to continue doing it. Now, let's go through the biliary tree. We have agreed that this is the liver. There's some ducts of bile which are within the liver. The ones outlined there. We correctly call them the intrahepatic bile ducts because they are bile ducts within the liver. And that means that the rest of the ducts we're going to talk about are therefore outside the liver. And so we can just call them the extrahepatic bile ducts. But there are many, so let's give them their specific names. The intrahepatic bile ducts join outside the liver, generally into two channels, one from the right lobe and the other from the left lobe. We call those two the right and the left hepatic ducts. So remember, they carry bile from the liver outside. The right and the left hepatic ducts join to form a common channel. And that common channel is what we call the common hepatic duct. The common hepatic duct is joined by the duct which come from the gallbladder. So this is the gallbladder, and this is the duct which is coming from the gallbladder. The duct of the gallbladder is known as the cystic duct. So this cystic duct join with the common hepatic duct so that they then form a common channel. That common channel is what we are calling the common bile duct. The common bile duct is joined by the duct from the pancreas. Usually the pancreas has two ducts, the accessory pancreatic duct and the main pancreatic duct. The common bile duct joins with the main pancreatic duct. Although they join, they don't in particular form another channel, not quite. They just open together. That point where they open together is known as the ampulla of Vater. It is in the duodenum. We also call it the major duodenal papilla. So this is the major duodenal papilla, or the ampulla of Vater. This is to distinguish it from the minor duodenal papilla, which is the opening of the accessory pancreatic duct, usually slightly above this level. Now, coming back here, the ampulla of Vater is surrounded by a sphincter. The sphincter that surrounds the ampulla of Vater is known as the sphincter of Oddi. We talked about it some time back. So this is the biliary tree. This is the path followed by bile. I want us to think through the possible routes or pathways of bile flow. And I want us to describe 3 possible pathways of bile flow. The first one is this. If you're not eating, and let me assume right now you are not, I might be really wrong, but it's fine. In a period of fasting, bile flows like this. So from the liver, it will go through the intrahepatic ducts, right or left hepatic duct, common hepatic duct, then to go to the cystic duct, then to the gallbladder. That happens when you are not eating during a fasting season. And the season doesn't have to be days, it's just hours, basically. So what does gallbladder do to the bile? The gallbladder will store the bile, but doesn't just store. It will also concentrate the bile and release that bile during digestion when needed. That then brings me to the second pathway of bile flow. When you're eating, when there is some food in the stomach and duodenum, and especially fatty food, what happens? Usually that triggers molecular mechanisms that will lead to gallbladder contraction through a hormone we call cholecystokinin. That hormone acts on the gallbladder to make it contract. And when the gallbladder contracts, we are going to define the second route of bile flow, which is from the gallbladder to the cystic duct, to the common bile duct, to the ampulla of Vater, to the duodenum. But there's a third way in which bile can flow. Bile can still come from the liver direct to the duodenum without going through the gallbladder. That's also a third route. So familiarize yourself with those 3 possible routes of bile flow. Histologically, this is how the gallbladder would look like. The wall of the gallbladder consists of a layer that contains smooth muscle. This smooth muscle enables it to contract when you need the bile and relax basically when you want to store bile. Importantly, the mucosa of the gallbladder is highly folded, as you can see. Highly folded mucosa to allow for stretch when gallbladder is filled with bile. But again, increasing the surface area over which bile concentration can occur. Told you that gallbladder stores bile, concentrates bile, and releases bile during digestion. So on the act of concentration of bile, the mucosa is highly folded to increase the surface area for absorption. And even the epithelium of the mucosa is simple columnar, usually with several microvilli. So what is the role of bile juice? Bile help in emulsification of fat. This is basically the non-enzymatic breakdown of large fat lobules into smaller lobules. Bile also help in the absorption of fat into the lacteal system. Physiologists will tell you more about what we call bile Michels and the like. Bile also contain bicarbonate, and the role of this bicarbonate is to neutralize the acidity of the stomach. remember the contents which come from the stomach to the duodenum are mixed with gastric acid, and so it will be of very low pH. The bicarbonate from bile has a high pH, and so it neutralizes the gastric contents that are entering the stomach, that are entering the duodenum. Bile is also a mechanism of excretion of waste products, which are basically excreted through bile, like bilirubin. All right, the fourth organ is the pancreas. I think we've talked about the pancreas before when you're looking at the endocrine system, so not so much news. The pancreas lies in the epigastrium as well as left hypochondrium. The tail is the one that extends towards the left hypochondrium. Usually it's located around the curvature of the duodenum, and that is primarily the head of the pancreas, which is located around the curvature of the duodenum. Both the duodenum as well as the pancreas are behind the stomach. The structures which are behind the stomach are known as being in the stomach bed. And when you come for practical, you'll appreciate this better. Anatomically, the pancreas has 5 parts. The tail of the pancreas, usually near the hilum of the spleen, within the left hypochondrium. The body of the pancreas is this one. This is the neck of the pancreas. The head of the pancreas is this one. Just below that part we are calling the head is a protrusion that we call the uncinate process. It could be part of the head, but it's unique, distinct, and so we give it its name, the uncinate process of the pancreas. Those are the 5 parts of the pancreas. Functionally, the pancreas has 2 components, the exocrine part and the endocrine part. Let's start with the exocrine part. The exocrine pancreas consists of pancreatic acini. So there are also acini of the pancreas, now pancreatic acini. The acini contain the acinar cells which secrete pancreatic juice, releasing into the ducts, which then join to form the main pancreatic duct, which take pancreatic juice all the way to the duodenum, opening together with the common bile duct, as you can see in this image at the ampulla ovata. The pancreatic juice contain digestive enzymes as well as bicarbonate again. This is how pancreas will look like histologically. It's a lobulated organ, as you can see, encapsulated, lobulated. You may easily confuse it with parotid gland, especially now that you are still seeing scattered adipocytes. You can still see lobulation. But there's something striking that you don't see in the parotid, but you see here. This region of light staining cells, a cluster of light staining cells. This is not an acinus. Maybe I show you how the acini look like. These are the acini. These ones are acini. And as we can see, they are serous acini. So you can easily confuse it with the parotid. But the game changer is on this one. There are regions where you have clusters of cells. They're not arranged in acini, they're arranged in a cluster. This cluster of cells, most of the time light staining, is what we call the islet of Langerhans. So this is the acini, which is constituting the exocrine pancreas. And this is the islet of Langerhans that constitute the endocrine pancreas. So as I've mentioned, the exocrine part releases pancreatic juice. The endocrine pancreas, as we mentioned back sometimes, is made up of the pancreatic islets of Langerhans, which secrete a number of hormones according to the different cell types. The alpha cells secrete glucagon, beta cells secrete insulin, delta cells secrete somatostatin, and the PP cells secrete pancreatic polypeptide. I hope you still remember the role of each of those hormones. Like glucagon was to elevate the blood sugar levels. Insulin lowers the blood sugar levels. Somatostatin is basically the one that inhibits pancreatic secretions. And pancreatic polypeptide is known to slow down food, although there could be other roles that are not clear. Now I'm done with the organs. Let's talk about the splanchnic circulation. The splanchnic circulation refers to the blood flow through the digestive system. So this is the model. We have what we call the aorta. The aorta gives you 3 branches which go to the digestive system: the celiac trunk, superior mesenteric artery, and the inferior mesenteric artery. The celiac trunk is the artery of the foregut, superior mesenteric that of the midgut, and inferior mesenteric that of the hindgut. The celiac trunk, therefore, supplies derivatives of the foregut, which include the stomach, the liver, the pancreas, but it also has a branch which goes to the spleen, in as much as spleen is not a derivative of the foregut. The superior mesenteric gives you branches which go to the derivatives of the midgut, which extend from the duodenum all the way to the transverse colon. And the inferior mesenteric gives you branches which go to the hindgut, which extend from the transverse colon down to the anus. Within those organs, wherever they are, whether foregut, midgut, or hindgut, there'll be capillary plexus around those regions. From those capillaries, you have veins. The vein from the GIT, the vein from the spleen, all meet to form the portal vein. Then the portal vein takes blood to the liver. The portal vein mixes with the hepatic artery within the liver. And as the David told you, the portal vein contributes the largest share of blood flow to the liver. So whether the blood is from portal vein, deoxygenated but having nutrients, and hepatic artery does not have nutrients but contain oxygen, the blood mixes within the liver sinusoids. And from the liver sinusoids, then we have the veins again, which go to the inferior vena cava. Those are the ones we call hepatic veins. There are usually 3, right, middle, and left hepatic vein. So that's the splanchnic circulation. But let's make our story complete by talking about regulation of the GIT functions. I know you've done this in physiology, but I like mentioning something just for the sake of completion as well. So there are those hormones which control the GIT function. There are also nerves which control the GIT function. Let's start with the hormones. The hormones which are produced by the digestive system, specifically from the wall of the digestive system, are called the gut hormones. They either come from the nerve terminals within the GIT wall or the epithelial cells within the GIT wall. Most of these hormones act on a paracrine action, and so you wonder why they're being called hormones. However, they also enter the bloodstream, and I think that's why we then call them hormones. They largely control the functions of the digestive system, although they may also have effects outside the digestive system. In our endocrine system, this is what we call the DNS, diffuse neuroendocrine system. The cells are scattered indeed, but if they were to be brought together, all of them, the endocrine cells of the digestive system will perhaps form the largest endocrine organ in the body. So there are a number of hormones with their specific effects. I've just picked maybe the 5 common ones. Gastrin, which come from the stomach, usually promote gastric acid secretion. Cholecystokinin, produced by the duodenum, largely promoting gallbladder contraction. Secretin is produced by the duodenum and jejunum. They act on the pancreas to promote bicarbonate secretion. GIP basically inhibit gastric acid secretion, and motilin stimulate gastric motility, peristalsis. There are many other hormones that you'll most likely been already told. If not, you'll be told in the GIT physiology. Let's finish the neuronal control of the GIT function. So the GIT is controlled by the enteric nervous system. There are two nerve plexus within the wall of the GIT, the submucosal plexus, which is also called the Meissner's plexus. It is located in the submucosa. That's why we call it submucosal plexus. The submucosal plexus are responsible for controlling GIT secretions, both endocrine and exocrine secretions. And then we have the myenteric plexus, which are located within the muscular layer, muscularis propria. Myenteric plexus are also called the Auerbach plexus. These ones control peristalsis. So whether submucosal plexus or myenteric plexus, these nerves are regulated by the autonomic division of the nervous system. Remember, this is either sympathetic and parasympathetic systems. You need to know from a physiological point of view the effects of sympathetic and parasympathetic divisions of the nervous system on the GIT. So first, let's distinguish the nerves responsible for the different functions. The parasympathetic effects are largely mediated by the vagus nerve. This is the 10th cranial nerve. The sympathetic effects are largely mediated by the splanchnic nerves. The splanchnic nerves come from the thoracic sympathetic chain, basically. On peristalsis, now this is how to distinguish. You don't have to cram this. Consider that you have two scenarios. In one scenario that we are calling parasympathetic, perhaps you are at home, you've overeaten githeri, and there's no exam soon. How do you respond? And so you try to look at how these parameters would respond in that particular scenario of rest and digest. Then on the other hand, imagine yourself, you are in this class like right now, then a cobra just appears in front of you. How would you respond? And the response here should not be a response of confusion. Should be a response that helps you to either fight or flee from the cobra. So if we look at it that way, then we'll see what should happen to peristalsis. When we've overeaten, peristalsis will have to increase. If you see a cobra, in as much as you may have peristalsis, and that's what I was telling you, don't look at the confusion part. The peristalsis when you see a cobra doesn't really help you to fight or flee, actually. As a matter of fact, sympathetic inhibits the peristalsis. But I know you may easily have diarrhea when you see a cobra. That is not a sympathetic effect. It's just a confused state because of reflexes. The sphincters relax under parasympathetic and tighten under sympathetic. If you increase peristalsis and you open the sphincters, then it means food moves very fast, and so you'll be having relatively loose stool. Sympathetic, on the other hand, if you reduce the peristalsis and you tighten the sphincters, means that food is not really moving, and so this person will actually constipate. Saliva is unique. Usually we talk of opposite effects of sympathetic and parasympathetic nervous system. But for salivary function, we don't talk of opposite effects. We talk of different effects. Parasympathetic system promote production of watery saliva. while sympathetic system promote production of mucoid saliva. Lastly, sympathetic nervous system inhibit production of gastric acid, while the parasympathetic nervous system through vagus promote the production of gastric acid. There's some patients who have a lot of acidity and they've tried other drugs and they're not working. Usually this is the last resort, vagotomy. It's a surgical procedure to just cut the vagus nerve that supplies the stomach so that it doesn't stimulate acid production. Great, so we'll stop there. We are done with what we're supposed to do today. Our next topic on the digestive system will be on development and congenital malformations of the digestive system. This is also a bit long, and so we'll do it perhaps in 2 parts. So we'll meet next week, same time, to talk about the first part, and then the other week, a second part. We'll stop there. I'll allow you to ask questions if you do have.